Kate Scow, and I'm representing soil microbiology once again. And today we're going to cover the lecture 10. You're actually getting 10 before 9. 9 will follow shortly, but this is lecture 10, Biodegradation and Bioremediation, Part 1. So what we're going to cover is a brief overview of environmental pollution and define what biodegradation means, and then talk about how microbes are engaged in these processes that end up in removal of organic compounds, or at least transformation of them, depending on the definition, and then give some examples of some applications of contaminants that are degraded through these different mechanisms by microorganisms. Then talk a little bit about factors that influence rates of degradation of contaminants, the chemical structure itself, the organisms, and environmental factors, which of course often rule in soil where availability is such a big issue, and this certainly applies a lot to contaminant degradation. Then in part 2, we're going to look more at bioremediation, which is the application of biodegradation. And the focus of this talk is going to be on organic contaminants, so on compounds that can be actually metabolized by microorganisms, leading to their transformation or destruction. So just to back up, risk assessment is a really important part of looking at pollutants in the environment, and I want to just show you where microorganisms and biodegradation fit into this overall larger conceptual view of risk assessment, which is what the EPA and other regulators use to decide whether or not a chemical poses a danger to humans and the environment, and how do they determine what are the safe concentrations of them. So in a risk assessment, you start with the source, like where are all these chemicals coming from? Let's say the chemical is, what's your favorite chemical? PCBs. So where do PCBs come from? Where are they used? Then how are they released? Where they're used? Does it go primarily into the air? Does it go into water? Does it go into soil? And then once it's in the environment, what are the transformations that it goes through? Not only transformations in chemical composition, but also movement of the chemical through the different compartments in the environment. That transformation and the movement are often mediated by microorganisms. So it's in particularly transformation where biodegradation takes place. That's one of the most important forms of transformation, because it can lead to the complete disappearance of a chemical. So it's very important. So that's where microbes fit. Then there's a whole aspect of trying to evaluate, so how much are people exposed to this contaminant, these PCBs, once they're in the environment and they figured out where they're going to go? What are the kinds of exposures humans are going to have? And also sometimes environment. People sometimes care about the environment, but it's more driven by human exposure. Are they drinking the water that has the chemical in it? Are they growing food in the soil? Are they breathing chemical that's like stuck to dust and flying through the air? And then from that, they'll estimate what is the uptake, like how much of the chemical are people taking up into their body. For example, when they shower with water, let's say it's contaminated with PCB, how much of the PCBs do they inhale in that shower, or how much goes across their skin, or how much will they eat in food if they eat a particular kind of food. And then that determines a dose of, then that goes into studies of what the chemical does in the body and toxicology studies. So once it gets in the body, what is the impact on the human? What's the kind of effect that it has? Does it cause cancer over the long term? Is there a short-term acute response? And then there'll be a dose-response curve that's developed for that particular kind of effect in the body for the chemical. And then out of that, there'll be a characterization of the risk. And that risk assessment is what the EPA has done for a lot of chemicals out in the environment that then helps the regulators figure out how to control emissions of the chemical or ban its use in particular kinds of applications and things like that. So that's the big picture, and you can see biodegradation is really important because it can make chemicals disappear. All right, and we know that contamination of soil and groundwater is a major societal problem, and it's becoming more and more of a problem as there's less and less of the resources. We're seeing that with water and with fracking and the need for using large amounts of water for recovering petroleum from places that it's not very easy to get out, and you have to pump a lot of water down in there to process it, and then there's not much water anyway, so what do you do with that water? Is that water, even though it might be contaminated, an opportunity for somebody to use? Or are you also polluting the water around where you're fracking? A lot of these situations going on more and more as there's just less and less resources to be able to use for this. Same with soil. There's a lot of contamination around industrial sites, contamination that's built up over time, and then what do you do about urbanization? What do you do about people who live in the vicinity of these sites? Often the people who live in those vicinities are poor, and often you've got environmental justice issues to deal with because, of course, the poor are who are going to bear the brunt of it. Similarly, groundwater contamination in the Central Valley, the little towns that have had their drinking water aquifers just wiped out entirely or private wells contaminated tend to be towns in which people who do not have a lot of income and buffer to get through such events live. So there's big environmental justice issues of it. So anyway, there's you can enjoy this picture as much as you want in your free time. Okay, so factors affecting the fate of contaminants. So when a contaminant enters into the soil, and that's shown here by OC, that's an organic chemical, so let's say it's a pollutant, let's say it's that PCB. So when it goes into the soil, it can undergo a number of different fates depending on the chemical properties of that chemical. So it can volatilize if it's got a high vapor phase, it can absorb into by plants, be absorbed by plants or earthworms or spit back out. It can adsorb onto clay or onto organic matter and be held in a form that's not immediately available for biodegradation, but of course if the chemical's passing through the soil and could leach to the groundwater, but it was held through absorption in an area that's biologically active, then you have more chance for it to be degraded. And then of course there's the Pac-Man, the microbes that can potentially degrade it, and chemical decomposition as well that may occur. And then you have water moving things out, but also water can move things back up to the soil profile. You can also have chemicals running off in runoff events. At the surface, there can be some photo decomposition. So anyway, my point being that you don't just have biodegradation going on in there. You have got to look at like all these different other processes that are going on in soil that may very much determine how much of that chemical is going to be available for biological degradation. So it's never just straightforward, just biodegradation in soil. You have to look at a lot of other factors. And these are properties of chemicals that are going to be very important in determining their ultimate fate when they go into the soil. Okay, some definitions. Biodegradation, biologically induced change in the structure of a chemical. And we have mineralization, which means turns to minerals, which is CO2 for organic compounds. It goes all the way to CO2, perhaps also other minerals if it has nitrogen or phosphorus in it. And also it will make cells if there's growth-based use of the chemical, right? But it's now going to be cellular material. That's the best way to biodegrade because it completely disappears. Another kind of definition is transformation, where there can be a change in the chemical structure of the contaminant, but we can't always guarantee that transformation makes it less dangerous. Sometimes it ends up creating a chemical that's more of a hazard to health and to the environment. So we have to be very careful about this definition of biodegradation, what exactly we're meaning by it. And people who are trying to sell products that they want to convince people are safe in the environment use this word biodegradation really liberally. And sometimes they really are only talking about transformation and not complete destruction. They say their product is biodegradable, but it may not be. It may just undergo some transformation. And then bioremediation is stimulation of microbial populations to degrade contaminants. And of course, I did that lecture on last Friday that used the case study of MTBE to show a lot of these kinds of things, kinds of definitions of bioremediation, which I will post that lecture, but that's another place to look for sure at this. Okay, so microbial processes that can influence organic pollutants. So here's just a big list, and we're going to show, showing in red are some examples of contaminants that undergo those particular kinds of microbial processes that influence organic pollutants. So the most straightforward one is primary substrate utilization, where the contaminant is a carbon and energy source for the microorganisms requiring an electron acceptor. Petroleum, many pesticides fall in this category. Co-metabolism is the transformation of the contaminant, but where it's used as a secondary substrate. The organisms that are carrying out that co-metabolic reaction that ends up degrading the pollutant are actually requiring also a primary substrate of something that isn't like a contaminant, an electron acceptor. And we'll look at an example of that and it'll make more sense. So chlorinated solvents like trichloroethylene is an example of that. Electron acceptor utilization, that's another way microbes use organic pollutants. We know in the lab we looked at perchlorate, which is not an organic pollutant, but that is used as an electron acceptor. But there's also other organic contaminants that can be used as an electron acceptor, thus needing a donor. And trichloroethylene is also an example of that. Then there's nutrient utilization. Some contaminants or pollutants that are actually pesticides, like atrazine, have nitrogen in it. And once people figured that out, they realized that they could set up conditions where it was nitrogen deficient, and that pushed the organisms to start using the atrazine to get the nitrogen out of it. So you can do interesting things once you know metabolically what's going on and why might the microbes use the compound. There can also be just physical accumulation of pollutants on the outside surface or inside microbial cells where they actually are like sorbents. And then also microbes can polymerize using extracellular enzymes that create these conjugation reactions that can actually bring together pollutants. And a lot of pesticides do this into the organic matter. They become feedstocks, these pollutants, for organic matter formation, if you can believe it. So that's an interesting thing. All right, primary substrate utilization. Now this can be both aerobic and anaerobic processes, and that's where we have direct use of the pollutant as a carbon and energy source. And a really good example of this is petroleum. But what's petroleum? Petroleum's not one thing. It's a mixture of a number of different organic molecules, including alkanes or aliphatics that have very short carbon chains to very long carbon chains. And the really short chain ones are really volatile and often will just evaporate right away when there's an oil spill. And the really long chain ones are ones that are going to hang around a long, long time, and they're difficult to degrade, whereas the intermediate chain length ones are the ones that degrade most rapidly. Aromatics are another important component of petroleum and actually degrade more slowly than the alkanes, but they're going to be both together in mixtures. And then also there's alicyclic compounds that will not go into much detail. Okay, high molecular weight compounds, so that means these really long chain ones, or aromatics that are multiple rings all glommed together, which you see sometimes, these polyaromatic hydrocarbons, they degrade more slowly than low molecular weight molecules. And a big, big issue for petroleum is availability, and because petroleum compounds are low in solubility, and they often, when you have a spill, they come out in big like blobs of a material that microbes can't really get a handle on. They need to be broken into smaller pieces to be able to get rapid rates of degradation, because otherwise the microbes just hang around the outside of it and degrade it as it diffuses. And we'll talk a little bit about that, but surface to volume limitations are a big deal with petroleum. And by the way, this leads to this really interesting stuff like of dispersive agents, dispersal agents, these other chemicals that have been added in oil spills to try to break the petroleum blobs down into smaller little blobs that microbes can break down faster. But there have been some big problems in using that. Maybe we'll touch on that, particularly in that more recent oil spill. I keep forgetting where it was. It'll come back to me. Okay. Oxygen supports the fastest degradation, but you get the whole sort of chain of events of going through the different electron acceptors when there is a petroleum spill. So other electron acceptors in anaerobic respiration can be used, and methanogenesis actually will eventually kick in, because there's fermentation also that will break down some of these petroleum compounds into simpler forms. Okay, so this picture here, well, this is just reminding you, and this is old, and there's so many more, but petroleum spills happen all the time, and they particularly happen because big ships carry petroleum around to different ports. They're leaving places where it's generated, or they're carrying them to other places. So there's been numerous spills all around. And there are also around pipelines some enormous spills as well. So we do have some soil events as well. This picture here shows you a gas chromatograph, which is showing you essentially a representation of all these different aliphatic compounds that are in a petroleum sample that's been shot into the gas chromatograph. And you can see there's all these different chain lengths. That's what these 11 to 26 shows you, these different compounds making up this mix of aliphatics. And then over time, these are going to slowly go down until finally you're going to see not many left. So this is just showing you a progression over time. And similarly, here's a mix of aromatic compounds, same deal, showing at the beginning of time, and then showing you with and without bioremediation. But just look at the changes in these peaks. You can see at the beginning there's a whole lot of different peaks, and they're quite high. And over time, they're going to be diminished as microbes degrade it, assuming they can degrade it. And actually, petroleum, a lot of compounds in petroleum are pretty degradable. The Exxon Valdez oil spill, I really should probably swap this out for the Gulf spill that was more recent. But this is a good example because there's history and there's enough time to understand what really happened. So in March '89, 13 million gallons of oil leaked from a boat up in Valdez, up on the Alaska coast, and just spread like this over all these islands. 1,000 miles of the coastline was coated, and there were massive kills of fish and sea animals. And I just wanted to talk about the biodegradation just to use this as an example. It's kind of a unique situation up there because it's very cold. So think of the constraints on microorganisms and their ability to degrade it. It's cold. The population densities of microbes are low because there's not a lot of carbon up in that environment where there isn't a lot of productivity, right? So there's not a lot of just background substrate to keep high populations present. Not a lot of nutrients as well. It's a classic oligotrophic environment. And then with the oil, you have this low surface to volume ratio. And there were a lot of studies done to test to see if adding microorganisms, adding surfactants, adding nutrients, using different combinations of it could help, like along these shores where all this oil washed up. And lots of research on that. And I think the consensus is that adding things didn't really help. It just didn't make that much of a difference in cleaning it up. Now, what did clean it up was bringing in like high pressure hot water sprays, and where they scoured the beaches. Here's one that's pure black and shiny with oil, and then they really scrubbed it down and made it really pretty and clean here. But in doing so, they ended up killing all the life, or a large portion of the life that was living there, and therefore just wiping out the ability of the system to deal with the contaminant through its own processes, even though they may be slow. So I guess bottom line is that there's a big debate over whether or not you should just leave oil there and let it, form these like tar balls and just slowly be degraded by microorganisms. Trying to add new organisms or add nutrients isn't really seeming like it's going to help very much. Maybe you just have to let it take its course, and it doesn't help with an engineering mentality to just go and wipe it all out and actually send it to the ocean. And just to say very briefly about surfactants, when people add chemicals that solubilize, and actually what they do is they make small micelles out of these like big blobs of oil, are broken down when you add these surfactants that coat the surfaces of these small micelles that are formed. And so you have then lots and lots and lots of little small spheres of the oil now, rather than it being in a big blob. And in theory, microbes could degrade it much faster. But I think what is being found is when you add those surfactants, it causes the oil to actually move away to other locations. And in fact, sometimes it's been found later that it looked like they've degraded, all those micelles were degraded, but what they ended up doing was sinking down into the sediment and accumulating in a different place. The other thing is some of those surfactants are quite toxic themselves and have their own problems for the environment. Okay, co-metabolism. So that was primary metabolism, and that was just an example of primary substrate utilization of a contaminant. Co-metabolism is really interesting. So it's the metabolic transformation of a substance, like a pollutant, while a second substance serves as the primary energy and carbon source for the organisms that are breaking it down. So what you do is you kind of trick an organism by feeding it what it wants to be eating, giving it what it needs, an electron donor and acceptor that it's used to having, and then you slip in the pollutant at the same time. And that pollutant is accidentally broken down by the enzymes that the organism's using to go after its primary energy source. And so the pollutant is fortuitously degraded by enzymes induced for other substrates which are normal. And this picture shows it here. Trichloroethylene is an example of this, a chlorinated solvent that is co-metabolized by a lot of organisms. So here's what the organisms want. These are like aromatic degraders that have this monoxygenase enzyme system. So they're being fed propane or whatever and breaking down to intermediates into CO2, just going on living their normal life, and you're slipping in the trichloroethylene. And that same enzyme complex will also break will transform the trichloroethylene into an epoxide, which is in a very unstable form of molecule, which just disintegrates then into CO2 and chloride. And you can see that's mineralization going on there. And it turns out that a lot of organisms can co-metabolize trichloroethylene. Anyone who has oxygenase enzymes, so that includes aromatic degraders, propane degraders, methanotrophs, like add methane, nitrifiers, really? Yeah, nitrifiers, you can add ammonium, and those guys can actually also break down trichloroethylene. What they all share, as I said, is these oxygenase enzymes that are being induced to degrade what the organisms want to be using in their normal lifestyle, but you can slip in these contaminants and they can degrade as well. So properties of it is there's no carbon and energy gain from breaking down the pollutant. Sometimes it results in dead-end products that aren't degraded further. In the case of the trichloroethylene, it seems to break down into harmless things. But that toxic metabolites might be produced, and for example, that epoxide being shown here, before it spontaneously degrades into these compounds. By spontaneous, I mean it just like chemically blows apart. But this has its cost on the organism that's carrying it out. Before it essentially disintegrates into these, it may actually be attacking the cell and hurting it. So there is some damage to the organisms that carry that out. The enzymes are often wide spectrum, right? They're not really specific enzymes. That's why they're promiscuous and they accidentally break down the wrong chemical. It's hard to study because you can't enrich microorganisms on the pollutant that you're wanting to study in this co-metabolic system because they're not growing on the substrate. But it's a pretty interesting kind of metabolism that is harnessed quite a bit in remediation. It's tricky because you have to make sure you have a good ratio of the primary substrate to the pollutant, because if you try to slip in too much pollutant in doing it, then they don't get enough of what they need, they don't grow, they don't repair themselves, so they end up eventually crapping out or dying. Okay, and there's been some examples of using this in field scale. In Savannah River, there was an enormous trichloroethylene spill. This shows the distribution of the trichloroethylene in this three-dimensional block at this site in Savannah River, which is at a national lab. And so you can see there's quite a bit of trichloroethylene there at these different depths in the sediment before anything started to happen. And what they did was they injected methane they targeted methanotrophs. So they injected methane, air for oxygen, nitrogen and phosphorus into the groundwater, into the contaminated zone. So essentially, infusing it with all these substrates that methanotrophs could use. And indeed, it stimulated the methanotrophs shown here, blown up, and they started to break down the methane along with the oxygen as the electron donor that was provided for them. But these are native organisms that were doing it, and they ended up co-metabolizing trichloroethylene, and that's shown here, that after I don't know, maybe I think it was 4 years or so, they got good removal of the trichloroethylene. So that's just an application of this understanding of microbial metabolism to the field. Okay, reductive dehalogenation is another way microbes use pollutants, and in this case, they're using the organic compound as an electron acceptor, and chlorinated solvents, again, are an example of it. So dehalogenation is one example of this use of a pollutant as an electron acceptor, where there's a removal of a halogen, often a chlorine, from an organic molecule. It's under anaerobic conditions, obviously, because it's used as an electron acceptor, and with oxygen around, it won't happen. And so this process is called reductive dechlorination. So here's an example showing you organic molecule with its chlorine sticking off on it. What happens is that it accepts electrons from a donor that's put in, and results with the chlorine being knocked off and if you have a multi-chlorinated compound with a lot of chlorines on it, then over time these will be knocked off as the compound accepts electrons. Remember I said it's an electron acceptor. Obviously you need to put an electron donor in as well to make that happen. Hydrogen is often used in applications of this process. And either depending on fermenters to produce it or adding it directly, but organic compounds are also used in this reaction. So what happens is that it ends up ultimately with the breakdown of the compound, and this shows you it here. Here's trichloroethylene with 3 chlorines on it, right? And it's accepting electrons. It accepts those. A chlorine goes off. Now it's dichloroethylene, accepts another one. Another chlorine goes off as it takes an electron, and then, uh-oh, you got vinyl chloride. Well, that's creepy, but hopefully it keeps going, and with some organisms it does, and another chlorine will go off, and then you end up having a very simple ethene molecule that's not chlorinated, that's really, really readily degradable. The problem is you can't always count on it happening. It can backfire on you, and there's actually a number of organisms that take it this far to vinyl chloride, and then it's no longer energetically as favorable to use. And vinyl chloride is a known carcinogen. Trichloroethylene is a suspected one, but we know that vinyl chloride is a human carcinogen. So if you end up with the reaction sort of ending here because the organism can't take it all the way, then you end up with a much worse problem than you started with. So something that has to be watched, and it's one of the reasons that bioremediation is sometimes not very popular with people, because they're afraid of situations like this. So we're going to look now at, in fact, so we looked at 3 kinds of ways that microbes use pollutants in different reactions: primary substrate utilization, co-metabolism, and as an electron acceptor with reductive dehalogenation. Okay, effects of different factors on biodegradation potential and rates. So when we're looking at bioremediation, we can't just look at the relationship of the pollutant with the organism, right? That's not in soil. You have to deal with the environment. So this shows you the 3 points: the substrate, the organisms, the environment. And then when you're looking at the relationships between each of the actors, we're talking about the biodegradability of the pollutant here with the organism interacting with the environment. Are all its physiological requirements being met? And between the substrate and the environment, is the chemical bioavailable and mobile? So this means we have to consider along this axis the volatility, sorption, solubility of the chemical. Along up here, looking at this relationship, we have to consider, why is the pollutant used? I mean, what metabolically is going on? And is the chemical toxic to the microorganisms? Or is it recalcitrant? Is it so complex that it just can't be broken down? And then we're talking about the organisms. We're wondering, are the right kind of organisms there? Are there enough of them? Do they have what they need? And so here, the organism can impact the environment by changing pH, physical structure, redox, which can then, in turn, affect this relationship between the substrate, the pollutant, and the environment, and also what the organisms are now living in as an environment now that they've changed it. And then the environment obviously affects the organism by providing nutrients, electron donors, acceptors, through the moisture in the soil and pH and other factors as well. But I'm just trying to get across to you that it's many interacting factors are going to determine, like I showed in that very early cartoon slide, that determines whether or not something's biodegrading. You don't just take an organism and a pollutant, put it in a flask and and study it and think that that in any way is going to represent what's going to happen in the environment. And believe me, a lot of people get money for studying. There isn't much money for biodegradation these days. I hope that changes and it comes back. But there's a lot of people who really are fixated on just genetically engineering an organism that will degrade a contaminant, and they just stay in the lab and, you know, work on fancy pathways or try to stick in genes. And then what do you have? You have some organism that is not going to be very capable of hitting the road and going out in the environment and making an impact with its great skills and talents. So here's some factors that influence the degradation of compounds. So these are very general relationships we're looking at. So we already know from the aliphatics that the chain length, like how big it is going to affect its rate of degradation, and also whether the molecule is folded on itself. Remember, lignin has a 3-dimensional composition that makes it hard for organisms to get access to. There also can be novel chemical bonds. This has been a big problem with traditional plastics that are human-made polymerization of different simple monomeric compounds that are very hard for organisms to figure out how to degrade. Also, large surface to volume challenges with a lot of contaminants. Sometimes this can be accelerated by the weather and weathering conditions or light that will start to like break down whatever is the issue, whatever is the kind of contaminant. In this case, if we use plastic as an example, that would be required to precondition it. And this shows you a fiber that is fresh in the environment and clean and very hard for organisms to get at. And this is after it starts to be weathered by the environment as well. And there's been a lot of interest in biodegradable plastics, and over the time that I've taught this course, it's really changed a lot, moving away from fossil fuel-based plastics to including material that may cause the molecular structure to open up a little bit and swell so that organisms can get in there, get access. And there's also plastics now made from microbial storage compounds like polyhydroxybutyrate that break down really fast. And those are some of the products that like the university uses for events, biodegradable plastics. Okay, and this is just a picture that's showing you plastic being broken down and what are some of the steps involved in it. So branching is another factor that determines how fast organic pollutants degrade. And here a really nice case study of this were detergents. When detergents were first starting to be used when I was a child in the 1950s, they created the first generation of detergents that went into washing machines, and they were made of alkyl benzene sulfonate, ABSs. Now the problem with that is the chemists who made that didn't think at all about what would happen out in the environment, because of course these detergents were going to be discharged to the sewage treatment plants and go out into the river. So what happened was that all those methyl groups that were hanging on alkyl benzene sulfonate made it impossible for microbes to gain access to these aliphatics and cut them right into smaller pieces and work to break it down. They just didn't have access to it. So they did some studies in the lab and actually thought about the biodegradability of the compound and came back with a new formulation of detergents, which were linear alkyl sulfonates that look more like this. And you can see there's just hydroxyl groups on it, and there was no longer this issue of access of microbes to the molecule, and they could break it down fast. And so then these detergents became much more biodegradable. The kind of substituent or thing hanging off the molecule matters a lot, and a really good example of this is chlorine. Here are two aromatic rings that are held together without any chlorines on it. It's biphenyl. With chlorines on it, in this case, two chlorines hanging on it, same thing, but with chlorines, it's dichlorobiphenyl. So the biphenyl breaks down quite nicely. It's easy for microbes to degrade it. They can access it. It turns into an aliphatic and an aromatic ring, and that goes into the TCA cycle without the chlorine. No problem. It breaks down. But if it's got the chlorines hanging on it, then it will break down into chlorinated versions of these two compounds, a chlorinated aliphatic acid and a chlorobenzoic acid. And these are much, much more difficult for microbes to break down due to steric hindrance by that chlorine being there, preventing access of enzymes to break it down. And so what you can have then is, though microorganisms might be able to take the first step using these oxygenases to partially break down, transform, actually, right, the molecule, the fact that those chlorines are there are going to prevent any further breakdown of the molecule, and you can have a dead-end product. So that's the kind of substituent matters a lot in chemical structure. And the number of substituents matter as well. Now this is really interesting. It's taking these chlorinated solvents that we were looking at trichloroethylene. Do you remember the figure in that earlier slide that showed the 3 chlorines hanging off of trichloroethylene, and there actually can be 4 per chloroethylene. So, and here's that dichloroethylene, and here's vinyl chloride, you know, the really toxic one that just has 1 chlorine. So what's interesting is under aerobic conditions, the less chlorinated molecules, the one with 1, has a much higher rate of degradation under aerobic conditions than the molecules with more chlorines on them. So the more chlorines you have under aerobic conditions, the slower, right? The slower the rate of degradation is. In contrast, under anaerobic conditions, the fewer the chlorines that are on it, the slower the rate of degradation is. The best electron acceptor, right? We're talking about anaerobic conditions, is the molecule that has a lot of chlorines on it and therefore has a lot of capacity to accept electrons and knock off the chlorines. But once you get down to not there being many chlorines on there, they are not actually energetically that favorable an electron acceptor. And so it's hard for the vinyl chloride to break down. And we saw that with some of the organisms that can't take the trichloroethylene all the way. So these opposite rules of thumb. Concentration matters as well. The concentration of the contaminant. This is super interesting because originally when people first started to do research, they would conduct their biodegradation experiments at pretty high concentrations of the pollutants, partly because we didn't have detection methods, analytical methods that could detect low concentrations. So we'd have to do what we could do, which was work at higher concentrations where you could use existing methods. And that's fine and dandy, and you can see, and here's a picture showing the concentration of a contaminant over time. It starts out at a high concentration and it degrades very nicely, looks almost like a first order, although first order doesn't work under really high concentrations, but it was high enough, right, that it degraded quite rapidly. And then people would say, oh, no problem, the contaminant will break down in the environment, it's not an issue. But it turns out for a lot of them, if you start at a lower concentration, actually the organisms cannot degrade it at all. So there's this concept of threshold concentrations for certain contaminants, that they can be at low enough concentrations that, in a sense, the microbes can't see them. And by that I mean is if there's going to be enzyme induction involved and needed to be able to kick in the reaction that would break down the contaminant, if it's at too low a concentration, there's not enough of the substrate to trigger the activation of the enzymes, and so it may just go like unnoticed and not degrade. So this issue of pollutant concentration can really make a difference. And then there's also the effect of organisms, and this is kind of a complicated slide, but it shows you a lot of things. It shows you some different pollutants here along this gradient, from pretty simple to degrade pollutants, phenol and toluene, which are aromatics, to naphthalene 2,4-D, to dibenzofuran, to fluorines, which are polyaromatic hydrocarbons, to TCDD, which is one of the worst. And so when you look at the frequency of organisms that can degrade these compounds, you can actually find like 1 out of 100 organisms in the soil will use these kind of compounds, but you get rare as you move in this direction, where you still have organisms that can degrade it, but they're not going to be as common in the environment, to super rare to we don't even know if there are organisms. So depending on the contaminant, it's going to determine whether or not there's any reason at all even to consider inoculating with organisms, like adding organisms. Like for these, it would be ridiculous to add organisms to break down things that there really is a lot of talent out there in breaking it down. And this also shows you that the focus of research at the level of the things that are really, these are the ones that are most abundant in the environment, are these widespread contaminants that actually a lot of organisms use, you'd be focusing more on environmental aspects. Like, what are the environmental constraints that are preventing it from degrading? Whereas with these more difficult to degrade compounds that we don't know if there's organisms or there's very few, you might focus more on molecular aspects and trying to develop organisms enriched for them, maybe transfer genes to develop them. But of course, I just want to remind you, as soon as you have that superorganism, then you've got to convince it to live in the environment, and it may have no ability to do so. So that's just the beginning of the story if you do find an organism or create an organism that can degrade a complex pollutant. Okay, inoculants are around. I just always say have a lot of skepticism, stay a critical thinker when you're looking at inoculants. There's a lot for sale. Particularly a lot of them for sale are for things like petroleum, for compounds that break down pretty readily by themselves. And there's all these products around BioPro, Petrobac, pH, Bacterial Consortium, the Mighty Bug Oil Eater. And I use the word snake oil because some of these companies really take advantage of the lack of knowledge that people have about microorganisms in the environment in general. People thinking that there are no organisms that have this capability, they must be added, or thinking that systems have gotten so depleted or damaged that you need to add organisms to make it work. But of course, if the systems are that way, like an ecosystem is really now reduced in any sort of life form, it's not an issue of just adding organisms back in because something has made it that way. There's some environmental constraints that are preventing the natural organisms and all the organisms that are adjacent to that site that would gladly move in at any time, ride dust, ride worms, follow plant roots into the area and recolonize the area. But there's something else that's keeping them from doing that because they would try. And so just adding organisms is often not going to be the solution, but it may sometimes. But anyway, there's just a big industry in this. These products are often very expensive, and just good to stay a critical thinker about this and think about the situations where they might be beneficial. So the success of inoculation varies greatly because you have a lot of issues when organisms that grow up in labs, in these cultures, and they're usually only a couple of kinds, Bacillus, Pseudomonas, the same players that wind up in all these inoculants because they're really easy to grow and they have a shelf life. They hang around when you put them in the product. But once they hit the soil, they're going to have to deal with predation and competition. Here's a really great cartoon that's showing these organisms coming into the environment to do their great job. Also, the organisms may not be in the right physiological state. They often they're going to have been grown up under really great conditions. They're going to be fat and sassy, and then they're going to hit hard times when they hit the soil. There may be things that the organisms need that aren't going to be present in the soil. A lot of times we add nutrients and also like an electron acceptor or whatever might be limiting the reaction along with the organisms. And then sometimes when you compare the inoculated with the electron acceptor plus nitrogen to just with the electron acceptor and nitrogen but not inoculation, there's not really any difference because what they needed all along was the nitrogen and the electron acceptor, not the organisms. And also access. How do you mix, particularly in soil, how do you mix the cells of your inoculum into the places where the contaminant is and where it can get access to the other things it needs to carry out the reaction? So it's tricky in a bioremediation context to get inoculation to work. In general, consortia work better than pure cultures in breaking things down. And this can be through commensalism, where there's organisms like co-metabolizers of PCB that produce these molecules. This one actually has a chlorine on it, like I said, that can make it slow to degrade. But in this case, there are degraders of this form that then can carry it all the way. There can be ones that can handle the chlorine. So this is commensalism. And also you can have detoxification. This strain BZ2 removes this metabolite that prevents this desulfonyl compound from being able to go further. So you can have somebody detoxifying the environment that'll keep the first organism in line to continue producing the product that the second one needs. So the effect of the environment, so this is, the whole course has been all about it. All those factors that limit rates of carbon turnover and also nitrogen, and the different processes we've been talking about throughout the course also apply to contaminants. And this includes, pH extremes, oxygen, nutrients, water content, all the things, all the same things that we've talked about throughout the course apply here as well. And then, as I keep saying, bioavailability is a major, major challenge in getting the organisms, the contaminant, and the other substances they need together in the same place at the same time. So this picture here shows you an example of reduced bioavailability. We have some soil here. We have these nice bacteria that are hanging around outside, and we'd be really happy to degrade the compound once it comes into the solution. And so this shows you a NAPL, which is a non-aqueous, it's a big blob of insoluble compound, and it needs to dissolve and come into solution. So that will be a process right there. You remember the two-compartment model, the two first-order model of kinetics? This would be that slower phase of this compound being dissolved and coming into solution, and then microbes could degrade it. Here's another bioavailability challenge of sorption, where the molecule is stuck to the surface, but it will slowly desorb, and then microbes will be able to degrade it when it comes out. So you have this concentration gradient around the particles where the contaminant is being held, that is due to the fact that anything that moves out further away is going to be degraded right away, but there'll be this sort of halo of material that's slowly becoming available closer to the surfaces. And some microbes have surfactants. This is cool. They actually release surfactants. Rhamnolipid is one example that the Pseudomonas aeruginosa produces that can actually be released extracellularly, often, again, with the organism having close contact with this pollutant, right, that's shown by this red squiggle that is not available. It's just in one big blob, but these rhamnolipids, which are just surfactants like detergents, are released that will then combine with the molecule and pull it into solution by making little micelles, and then the organism can degrade it. So it's kind of cool that there are such types of capabilities out there, and people are interested in these surfactants because they presume they're safer than surfactants that we create in industrial processes. And so interest in harvesting these surfactants in bioreactors and using those in the environment. Okay, and then the last issue is this process. This is such an interesting situation. Ethylene dibromide was a chemical that was used for a long time. It's very volatile. It was used for fumigation for many years, but then they realized that it was quite toxic. and stopped using it. This is over in the East Coast. And this picture here shows you a soil. There's two different soils, but we can just look at soil A. And here's the native ethylene dibromide. This is left over like 20 years after it was used. It was still present. It was still hanging around and not degrading. So you could take some soil and put it in the lab and incubate it over time, and the concentration didn't change at all. However, if you added fresh ethylene dibromide, which is really actually quite degradable, so you could add it in the same soil, and it would degrade right away and then completely disappear. So you had this big difference between recently added ethylene dibromide and this native stuff that was around a long time. And they found that if they took a mortar and pestle and ground up this soil that had the ethylene dibromide in it, they could actually get it to degrade. Not all of it, but they could get more of it to degrade. So this led to this realization that there was this other kind of process going on that made molecules not available, not just through sorption or being insoluble, but these were trapped, physically trapped in small pores inside clay and organic matter, and could only very slowly diffuse out to microbes, or basically not at all. Some of them were trapped in dead-end pores. So then it comes into this interesting discussion of, does it matter them if they're so trapped? We had this discussion earlier in the quarter in the class about it as well. Is that fine? The fact that there's this ethylene dibromide that we know is toxic, and it's present at this concentration in soil, but microbes can't get at it. Can anyone else get at it? Well, you can ask, can worms get it? And so people do studies putting worms in these soils. Did the worms take it up? Maybe a tiny bit, maybe not much. Can plants take it up? Maybe not so much. How about if there's a big storm event? Does it push it out and it moves to the groundwater? Well, it doesn't really seem to move that much there. Well, what if you had this big catastrophic event, like this kind of situation where there was a lot of disturbance in the soil? Could it then become available and cause a problem? And so those are the kinds of discussions and considerations that regulators have to go through to think, do we need to treat this soil that's got this chemical that we don't want there? Do we need to treat it because one day maybe that chemical will become available? And if so, how will we treat it? Because actually what they'd have to do is dig it up and burn it, I think. I don't know how they could actually treat it. Or do we leave it here because we know that it's not going to go anywhere? And can we believe that? So anyway, interesting challenges because microbes can't get access to some of the chemicals. So we're done, and I'll carry on with the next part of this lecture.